Yeah, so my name is Cheri. I'm here with Cameron to tell you about this new technology that we developed that enables us to perform massively multiplexed nucleic acid detection using Cas13. And just to reiterate that introduction, everything that we're going to tell you about today was done at the Broad Institute through our roles as postdocs. And that's important to mention because both of us are in the middle of these career transitions. I left my postdoc a couple of months ago to found Concerto Biosciences, and then Cameron will be leaving his postdoc in January to start a faculty position at Princeton. We are both fundraising and hiring. So if you want to partner with us financially or work on this technology or similar technologies, please don't hesitate to reach out. We would love to chat with any of you about that. And so with that, I will hand it over to Cameron for the scientific introduction. Oh, crap, sorry.
Yeah, definitely. Thanks, Cheri, for that. And yeah, normally this is a slide that I have to really build up, but I think on a day like today, we can all agree that detecting viral infections is really important. But I did want to break down the problem a little bit just in terms of throughput versus multiplexing. So what do I mean by that? Multiplexing, the number of different viruses or targets we might want to detect, whereas throughput is the number of samples to detect at a time. And there's often a trade-off between those two. So at the moment, for example, we really care a lot about one virus in particular. We need to test a lot of samples. But in other circumstances, we might want to test for a larger number of viruses. For example, if there's a patient who is sick and we don't know what they have. And then there's disease surveillance, which sort of falls somewhere in the middle. We can go to the next slide, maybe. And so existing technologies, unfortunately, generally only allow you to test a small number of samples for a small number of targets at a time, as you can see by these various dots on the graph. And many of them are limited by the size of a conventional 96-well microtiter plate, which is that black line that I'm showing you. And unfortunately, if we overlay the scale of the problem on top of that, we notice that many of these technologies really fail to reach the scale and ease of testing that's needed to solve these problems. And so what we really want to do is move up and to the right on this graph. And Cheri will tell you just shortly how we plan to do that. But first, let me tell you about SHERLOCK, which is the CRISPR-based detection technology that underlies CARMEN. And the way that SHERLOCK works is you start with a DNA or RNA sample, you extract nucleic acids, you amplify those nucleic acids, convert that amplified double-stranded DNA to RNA, and then use Cas13 to detect that RNA. And Cas13 is this really special CRISPR effector protein. It's sort of like Cas9, but it binds to RNA instead of DNA. And once it does bind to its target, it becomes really active, and it can cut a reporter molecule many, many times after a single specific binding event. And that's really powerful because it amplifies that individual specific binding event into many, many subsequent cleavage events, and it really amplifies both the sensitivity and the specificity of diagnosis. And a standard SHERLOCK reaction is really one sample with one detection mix. And a couple of years back, our colleagues in Feng Zhang's group showed that you could do 4-plex multiplexing with Cas12 and Cas13, but we really were a lot more ambitious. Our goal here was to enable 100-plex multiplexing with many, many samples in parallel. And Cheri will tell you exactly how we did that.
Thanks. Yeah, so to do that, we invented CARMEN. And before I tell you how CARMEN works, let's talk about the underlying principles that make CARMEN possible. So the first one is miniaturization, which means that we take reactions that we normally would perform on the microliter scale, and now we're performing them in nanoliters. And this means that we can now do thousands of reactions in the same space and for the same cost that you would normally need to do a couple of hundred reactions. And then the second principle is self-organization. And this means that instead of setting up each reaction in a deterministic way, so we're going to take patient sample 1, and first we're going to test it for COVID, then for flu, then for RSV. We're going to take all of the samples and all of the detection mixes that we're testing and let them randomly associate. And in this way, we can form thousands of reactions in parallel in just a few minutes. And so the end result is a technology that can perform 4,500 tests in parallel. It does this comprehensively, and it's cheap enough that now we can actually practically scale it to test millions of people. So how does this work? We start by preparing the patient samples by isolating nucleic acid from the sample and amplifying it using multiplex PCR. And then we're preparing the detection mixes separately in parallel. To each sample and to each detection mix, we add a unique color code. This will allow us to track the location of each sample and detection mix during the experiment. The color code is like food coloring, except that it's fluorescent. And we make the color codes out of ratios of fluorescent dyes. And this allows us to make over 1,000 different color codes. We then emulsify each solution into nanoliter droplets. And we pool the droplets together. So now in a single tube, we have all of the samples and all of the detection mixes, but they're not interacting yet because they're partitioned into nanoliter droplets. And the color of each droplet tells us what's inside the droplet. So now what we want to do is make all possible pairs of each sample with each detection mix. And to do this, we flow the pool of droplets over an array that contains thousands of microwells. And just if you can see my video, I'm holding one of them in my hand. It's this flexible PDMS for those of you who do microfluidics or fab. It's this little plastic device, and this has just over 40,000 microwells on it. And then I also have another one with me. So this is the larger version that we make. It's about the size of a cell phone. And this one has just under 200,000 microwells on it. And each of those microwells is the correct geometry to catch exactly 2 droplets. So 50% of the time, we're gonna get 1 sample droplet and 1 detection mix droplet. And then when all of the wells are loaded and the excess droplets are washed away, we seal the chip and use fluorescence microscopy to read the color codes. So now we know which sample and which detection mix ended up in each microwell. We then apply an electrical pulse that merges the droplet pairs within each well, and this initiates thousands of detection reactions simultaneously across the chip. And then we can monitor those reactions over time using fluorescence microscopy. So for any well where we see the reporter fluorescence turn on, then we go back to the color code map that we made at the beginning, and we know that that sample is positive for the virus that that detection mix was testing for. Okay, so what does the data actually look like? This is, at the top, you can see some fluorescence images of the color codes and the reporter channel before and then after droplet merging. So after merging, we quantify the reporter signal, and you can see below this plot of the fluorescence intensity is on the y-axis, and then concentration of the input DNA is on the x-axis. And this was just for a simple limit of detection experiment that we did with Zika virus as an example. And high concentration is on the left, and then the concentration goes down, going to the right. So you can see that the sensitivity of Carmen is on the order of single attomolar, which is 1 copy per microliter, which if you think about that is pretty insane. And one thing to note here is that each sample detection mix pair appears on the chip in many different locations. And so that's the distribution of values that you see here. And because we have many replicates of each pair, we can get these statistically robust results, which are essential for the excellent performance of this technology. However, visually representing a matrix of distributions for every experiment is super difficult. So when we display data, what we actually do is take the median and plot that as a heat map, which you can see down here. So every time that you see a heat map in this presentation, remember that what you're actually looking at is the median value. I keep losing my mouse, there we go. The median value of the distribution of replicates for each sample detection mix pair on the chip. And then lastly, we can ask how well the assay performs in droplets versus on a traditional like 96 or 384 well plate, which is what Cameron and Fung's lab had been doing previously, and the answer is the same. So you can see in both plates and droplets, we get attomolar sensitivity, which is great. Okay, so now we have Carmen. What are we gonna do with it? We demonstrated Carmen in a series of increasingly difficult nucleic acid detection problems. So first we identified different viral species. Then we showed that we could identify subtypes within a single species. And finally, we demonstrated that we can detect individual genetic mutations, or SNPs, single nucleotide polymorphisms, that can make a virus drug resistant. And so we're going to tell you a couple of vignettes about these. The first is species detection. So I don't know about you, but when I have a technology that could possibly be comprehensive, I get a little competitive and want it to actually be comprehensive. So we thought, could we design a panel that would test for all human-associated viruses at one time? What would it take to do that? So to do that, we pulled all of the genomes for every virus that had at least 10 published genomes in October of 2018, and that was 169 viruses. The reason we need at least 10 genomes is because you have to have a nucleic acid sequence to be able to design primers and CRISPR RNA, and there's kind of no point in designing against one sequence because you don't actually know how representative that sequence is of the other strains of that virus that are out there in the world. So we decided to use 10 as a cutoff here. So using those genomes, we designed PCR primers and CRISPR RNAs, which could amplify and detect each virus. And then we used CARMEN to physically test the performance of each of those assays against every single target. And that was over 28,000 tests. And then our goal for this was that this collection of tests would be a modular master set of tests, so that when clinicians or other scientists want to test for a subset of viruses that are specific to, say, a certain geography or a certain body fluid, the tests are already designed. And you can just pull them off the shelf and put them in whatever configuration you want and use them. So we did that, and it worked really well. So on the vertical axis, you can see all of the different CRISPR RNAs, and we haven't labeled them all because that would be really hard to read, but you can see the viral families for each of these CRISPR RNAs. And then on the horizontal axis are all of the synthetic targets that we used, which represent each of the human-associated viruses. And you can see that 94% of the time, the CRISPR RNA detects its intended target and nothing else. And that forms this line that you see on the heat map. And since each CRISPR RNA performs independently of all the others, we can assess the CRISPR RNA performance individually. So for each CRISPR RNA, we calculated the area under the curve for a ROC curve, and the median AUC out of all of the CRISPR RNAs was 0.997, which indicates excellent performance. All right, so synthetic samples are nice, but what ultimately matters is how well this technology performs in patient samples. So we got a hold of 58 samples from patients that had known infections, including dengue virus, Zika virus, influenza, HIV, and hepatitis C virus. And these patients are here on the vertical axis, and then the different CRISPR RNAs are on the horizontal axis. And the trouble of doing comprehensive testing is then you get comprehensive data and you have to figure out how to display it. So we're just gonna walk through this step by step. The first question we can ask is, do we see the expected infections? And the answer is yes. So you can see by the green arrows that we detect dengue virus in the dengue samples, we detect Zika in the Zika samples, and so forth. Okay, that's great, but like, what is all of this and what's all of that? Is this the presence of a real virus? Is this cross-contamination? One way to figure that out is to compare these results to another comprehensive, although much more expensive detection method, and that is metagenomic sequencing. So when we did that, we see that the majority of the unexpected viruses that were detected by CARMEN are also detected by sequencing, suggesting that these viruses were actually present in the samples all along, but the clinicians or the people who were doing the testing couldn't see them because they were using these single target tests. And then finally, we can ask if the presence of these viruses makes biological sense, like just because sequencing can see them, just because CARMEN can see them, like should they even be there? And what was kind of cool is that the answer is yes. So for example, Peggyvirus A is a common co-infection with HIV, and we only observed that in HIV samples. And TLMV, which is Tortenol-like minivirus, is a very common blood infection. It doesn't have any symptoms, so nobody really knows if they have it. And we see it only appearing in the blood samples. We don't see it in the flu samples, which are throat and nasal swabs. So then if we kind of zoom out and compare CARMEN and sequencing overall, we performed over 11,000 tests in order to test these 58 samples. And the vast majority of these tests yielded interpretable results for both CARMEN and NGS. And then within these interpretable results, 99.7% of the time, CARMEN and NGS agreed. And then where they disagreed, which is here and here, it's pretty split evenly between the cases where CARMEN found something, gosh, my mouse, there we go, where CARMEN found something and sequencing didn't, or sequencing found something and CARMEN didn't. And then finally, I mentioned that our goal really was that we wanted the human-associated virus panel to be this modular master set that we could use to assemble situation-specific panels. Little did we know that COVID-19 was coming, but when it did show up in a matter of weeks, we were able to pull, so this is just a small bit from that giant square I showed you earlier. When it showed up, we pulled the coronaviruses out of that master set. We added a test for the novel coronavirus, and then we were able to make this coronavirus specific panel. And since then, we've been working to adapt this technology to the pandemic, and Cameron will tell you more about influenza and a respiratory specific panel in just a minute.
Yeah, that's right. And so as you can see from what Cheri has shown you, CARMEN works really well for species detection. We really wanted to challenge ourselves though and see how sensitive and specific the Cas13 detection could be by going after both subspecies and SNPs. And to start, we decided to go after subspecies, and the canonical example here really is Influenza A, which is the causative, one of the causative agents of flu. And it is canonically divided into many, many different subtypes based on the hemagglutinin protein and neuraminidase protein. And there's over 16 H subtypes that have been described, H1 through 16, and 9 neuraminidases, N1 through N9. Now, some of these infect humans, others infect other species like birds. There's even, for those of you who are flu aficionados out there, a specific type of flu that infects tigers and is apparently very lethal in them. So watch out if you're a tiger. That was the problem we set out to solve. And so we decided to make a CARMEN assay to try and detect each of these different subtypes of flu. And as you can see, the results here really speak for themselves, I think. When we tested this on synthetic targets, we could see really good performance differentiating all of these different subtypes, H1 through 16 and N1 through 9. We also tested this on patient samples and saw really good performance here as well. This is a set of 20 throat or nasal swabs that we obtained from collaborators at CDC, which were either H1N1, which is the first set of samples, or H3N2. And I think you can pretty clearly see, as Cheri is showing with the mouse, which ones are which. And this lined up with 100% concordance with both quantitative PCR assays for subtyping as well as next generation sequencing. So we were really excited to see the performance in that way. We've also done some really nice work with SNP discrimination that unfortunately I don't have enough time to really talk about in detail. There we went after a number of different drug resistance mutations in HIV reverse transcriptase and integrase. I believe it was a total of 27 different mutations, and tested that on a set of patient samples. But what I really want to talk about is actually some of our more recent work. This is all unpublished, so please don't share this outside of this group. We really wanted to show you some of the latest and greatest stuff that we're doing, but it is very early stages. And so we've developed a CARMEN respiratory panel that can go after 10 different respiratory viruses, shown here in this heat map. Obviously, SARS-CoV-2 is on there, as well as influenza A and B, and a number of other related viruses. And what we're just showing here is a simple limit of detection experiment where we're diluting each of these viruses down to different concentrations and showing really quite strong performance across the board here. And this was really exciting to us because this is one of the reasons why we developed CARMEN in the first place, was to be able to go after a problem like this and quickly design and test and iterate on a panel that performs really well. And indeed, we tested this panel on a set of patient samples. I'm showing you here just a subset of the sample testing we've done on a set of flu samples as well as COVID-19 samples. And what you can see here is we can detect flu A as well as SARS-CoV-2 with really good performance, as well as high concordance with other assays that were performed to determine whether or not the virus was present in those samples. And in that case, it was a quantitative PCR assay that was performed. So we're really excited about this technology and to use it more going forward. I think there's a lot of use cases here, which I'll sort of talk about in this slide, which is really that we need testing technologies that scale if we're really serious about reopening the world and having lots of people interact with each other. And that could be at the workplace, at an airport, in a school, or goodness, in a stadium. It's really hard to imagine these days that many people getting together in one place at one time, but hopefully someday we can get back there. And unfortunately, a lot of the existing technologies don't really get there. I'm plotting these axes in terms of turnaround time and batch size, because the goal is really to have a test that can be done quickly and that can let you test lots of samples at once. That's the only way to scale to solve this problem. And unfortunately, many existing technologies solve one or the other, but not both simultaneously. So take sequencing, for example. It's a great technology. You can test potentially lots of samples at once, but the turnaround time is pretty long. And so it's hard to imagine that technology being used to give people rapid results, especially in something like, say, a drive-through testing facility. And conversely, there are some other methods that have been developed that you might have heard of, like the Abbott ID Now or Cepheid Gene Expert, that have a really quick turnaround time, but unfortunately, they've been designed for testing individual samples at a time, and the throughput there is really limited. Each machine can only test a single sample at a time, and so a single machine can only test a few dozen samples a day, and that really doesn't cut it given the scale of the problem. And so the goal is really to combine these two things. What we've been talking about today has mostly been CARMEN, which I think has potential to move to the right on this graph, and that's something we're actively working on and I'm really excited about. But in some of my other work, we've also been pursuing more point-of-care detection strategies based on CRISPR technologies like SHERLOCK, with the goal there of making those more scalable, so that they're currently optimized for testing small numbers of samples with rapid turnaround. We'd like to see them get used also for testing at scale. And with that, there's lots of people to thank, and I think Cheri's going to be joining me on this slide, as I know there's so many people that were involved in this project in different ways. I'm particularly grateful to Pardis, who's been a phenomenal mentor throughout my postdoc, as well as Catherine Friege, a really talented graduate student in the lab who I've had the privilege to work closely with for a number of years on this project and many others. Cheri, you want to jump in and thank a few other folks?
Sure. Yeah. I would really also like to thank Paul, who has supported this project completely. And it was kind of outside of the Blaney Lab wheelhouse to go into diagnostics, and he took it on completely, and it's been great to work with him. And then Gautam Thakur, who is a graduate student who's joint actually with Paul and Deb Hung, was also instrumental in getting this off the ground. And these are all of the funding agencies that have supported us along the way. We are especially grateful to unrestricted funding. So for those of you who support academic research in a way that allows adventurous research, which the Hertz Foundation certainly does, thank you so much, because technologies like this don't exist without funding sources like that. And then obviously feel free to reach out. Our next email addresses are there, so those will be good for a while, and you can always find us on Twitter.